Module 5, protein. So what are proteins? The word protein is derived from the Greek word proteos, which means primary. It consists of amino acid molecules, which are considered the building blocks of protein. So protein is going to contain organic molecules, which means that it's going to contain carbon. We'll have oxygen atoms. It's going to have hydrogen atoms as well as nitrogen atoms. The amino acid structure is very unique to amino acids and protein, but there's a commonality among all amino acids. And I'd like to point your attention to the bottom right-hand corner of the screen, and we'll talk about this. So all amino acids are going to have this centrally located carbon atom. They're also going to have this hydrogen atom. They're also going to have this carboxylic acid group, or the COOH group, or the acid group. And they're also going to contain the amino group, or the NH2 group. And this is really what makes protein so special. The human body has a need for nitrogen, and it's nitrogen that makes amino acids so special. So we need to consume protein really for the nitrogen that amino acids contain. Now, all amino acids are going to contain this structure, but what makes them different among each other is this blank white square that you see here, which represents the side group. So there are various structures that are going to hang off of this centrally located carbon atom, and that's what differentiates one amino acid from another. So they are going to vary in size, they'll vary in shape, in electrical activity, as well as pH, and it's the side group that confers these particular traits. So how do we get these amino acids to link together and break apart? Well, we use condensation and hydrolysis reactions. So as you recall, condensation reaction is where we draw water from the two amino acids that we're putting together, whereas hydrolysis, we're using water to split those two amino acids. And so this diagram at the bottom of your screen here is basically showing two amino acids being put together, where we use a condensation reaction and we pull off a hydroxyl group and a hydrogen atom here. We'll bring those together and we draw that water out and basically form the peptide bond and form this dipeptide. So a peptide is just another way of saying protein. And so in this instance, since we put together two amino acids, we have formed a dipeptide. Protein comes in various structures, and the very simplest form of a protein exists as a primary protein, the linear sequencing of the polypeptide chain. So this is basically a whole bunch of amino acids put together in a single line. So we just go right down the line and we added one amino acid after the next. We can also have protein exist as a secondary structure where it's folded or coiled upon itself. Basically all these polypeptides arranged in this manner. And the alpha helix of DNA is an example of this. The beta-plated sheath as well as random coiling of proteins. A great example of this being collagen. Collagen has a secondary protein structure that is the foundation for a lot of things like bone and blood vessels and skin, etc. So we have to have collagen there as the foundation of protein in order to build stuff upon that collagen. Protein also exists in a tertiary structure. It takes on a 3D shape. It's globular-like. Enzymes and transport proteins and immunoproteins typically have a tertiary structure. So we're getting more and more complex here as far as the structure is concerned. Protein also exists in a quaternary structure, meaning that it will have several components put together to form one unique protein. The best example of this in the human body really is hemoglobin, which is depicted here in this picture in the bottom right-hand corner of your screen. Protein has 4 subunits, represented here by the 4 different colors that you see, put together in order to make hemoglobin. So that's a quaternary structure. So where do we get proteins from? Well, the exogenous sources of protein are fairly ubiquitous in our diet. We get them from both animal and plant sources. So it's pretty easy to get protein in the diet. We also produce proteins within the human body. So this is endogenous production. Every about 3 to 4 to 5 days, we replace gastrointestinal tract cells. And so we have to decommission those, and we call those desquamated mucosal cells. So we lose about 50 grams per day of protein this way by simply decommissioning these cells and then removing the components of the cells. And so we lose some protein this way. Digestive enzymes and glycoproteins, particularly with the enzymes, every time we have a meal, we're sending in enzymes to go in and digest our macronutrients, and we're producing protein this way. So we'll be eliminating about 17 grams per day of protein in this manner. So how are amino acids classified? Well, there are several ways to do this. We can classify amino acids by their structure, so simply by that side chain that we talked about earlier, and that's depicted in this picture down here in the bottom right hand corner of your screen. We can also look at the net electrical charge, and we can also look at the polarity of whether or not it's polar or non-polar. If you recall from your molecular biology lessons and chemistry lessons, a polar molecule interacts nicely with water. It's hydrophilic, whereas a non-polar molecule doesn't interact with water nicely. It's hydrophobic. So the polarity can also be a way to differentiate one amino acid from another. We can also categorize proteins as far as their essentiality. So whether or not they're essential nutrients or non-essential. We can further subdivide these into conditionally essential. Conditionally essential just means that there may be a disease state or some sort of disorder that makes a particular amino acid essential when in a normal, healthy person, you wouldn't have that amino acid be essential. So you wouldn't have to ingest that particular amino acid. We could actually produce it. But there are conditions, diseases, disorders, etc., that would force that particular amino acid to become now essential. So this is a table showing you how to classify amino acids based on all of the things that we just talked about. So you've got essential and non-essential. So the essential amino acids we would have to ingest in order to meet our nutritional needs. And we can then subdivide that based on its side chain, the charge that it contains, and the polarity that it contains. And we do this both for essential and non-essential amino acids. This little footnote here with the stars, that just means that isoleucine, leucine, and valine, those are the branch chain amino acids, which we'll talk about later on in the course, and which have implications for athletic performance. So, what do we need proteins for? Well, this long list here basically shows you that what aren't we using protein for, essentially. But protein are the foundations for enzymes. I mean, any enzyme that you can think of, whether it be for energy production or to, metabolism, to repair tissues, etc., hormones, these are all protein-based. So hormones like insulin, glucagon, epinephrine are protein-based. It could be structural components, bones made out of protein, ligaments, tendons, muscles, organs. The immunoproteins, so our immune system cells, many of those are protein-based. To maintain fluid balance, albumin is a visceral protein that's very, very important as far as maintaining the plasma portion, or the watery portion of blood within blood vessels. So it's maintaining what we call the colloidal pressure in blood vessels. And when you don't have enough protein or albumin in the bloodstream, you could end up with a displacement of that fluid, particularly into the peritoneal cavity. Just as an example, although this particular infant may not be suffering from albumin, but it's just an example of how protein, or the lack of protein, could lead to edema or fluid accumulation like this infant in the bottom right hand corner of the screen. So it's important for fluid balance. Of course, protein does provide us with some energy, 4 kilocalories per gram. We can also use proteins to transport stuff. We talked about hemoglobin. There's also myoglobin, the analog of hemoglobin in muscle tissue. We can use it to transfer substances between cells. And of course, albumin is a transport protein as well, other than being important for fluid balance. So it's very, very important as far as protein is concerned that we are using protein in all kinds of manners in the human body. One interesting point about protein is also that we use it as a buffer. So because of the electrical nature of amino acids, we can use it to mitigate pH changes in the blood. So when the pH is too low in blood, we can actually use amino acids to accept hydrogen ions to increase the pH a little bit more, bring it back within normal limits. And conversely, we can do the same when pH is too high. We're going to donate some electrons to, or hydrogen ions, to reduce the pH when it's too high. Some other roles for protein in the body, well, we can produce what are called conjugated proteins, and these are just proteins that are joined together with other components that are not made out of proteins. Glycoproteins are an example of this. It's basically glucose plus a protein. Intrinsic factor, which is a protein that's required by the gastrointestinal tract in order to absorb B12, would be a great example of this. We have proteoglycans, which are proteins plus long chains of disaccharides or carbohydrates. We have lipoproteins, which transport lipids but have a large protein component to them. There are also flavoproteins, which are these protein enzymes that are bound to things like flavin mononucleotide or flavin adenine nucleotide. These are vitamins, essentially, riboflavin being the flavin mononucleotide, that are essential for chemical reactions in the human body as far as those nutrients are concerned. We also have metalloproteins, which are basically a protein plus a mineral. So how are proteins digested? Well, as with all the macronutrients that we've discussed, when we eat food, we begin in the mouth with mastication. So we've got the physical phase of digestion. We should note that, even though I don't have it listed here, that saliva is released into the oral cavity to begin moistening and lubricating the food, and as well as providing some immunoprotection against any possible antigens that are on that food. When we hit the stomach, we continue with mechanical digestion. So we've got the churning that occurs from all of the muscles contracting in the stomach, which allows that acid, that chemical part, that hydrochloric acid, to be mixed in with the partially digested food, which then will break apart these long chains of peptides into smaller and smaller bits. The point of the digestion of proteins is to get down to the level of amino acids and some dipeptides and tripeptides. So the hydrochloric acid is going to perform what's called denaturation, or it's going to denature the structure of protein. So it's going to take those complex structures that we were talking about, the quaternary, the tertiary, and the secondary structure, and basically unwind those proteins into more of a primary structure, so that then the enzymes can break apart the bonds that are keeping the amino acids together. So there are some enzymes in the stomach that are released. Particular pepsin, again, is going to break apart these peptide chains into much smaller ones. And it's responsible for about 10 to 20% of protein digestion. So that's a pretty significant amount. Of course, the goal here, again, is to free up some of those amino acids. But in the stomach, basically we're getting just some peptides, polypeptides, oligopeptides, and it's really in the next phase, the small intestine, where we're going to see a significant amount of these free amino acids floating around in the GI tract. We also, of course, release mucus to protect the lining of the stomach from the hydrochloric acid. In the small intestine, we're going to get secretin and cholecystokinin secreted from the mucosal sites to stimulate the pancreas, and it's going to stimulate the pancreas to release bicarbonate, which is going to protect the small intestine against any acid that was mixed in with the partially digested food from affecting the unprotected lining of the small intestine against the acid that was mixed in with that partially digested food. We also are going to release some electrolytes and water. Now, as far as enzymes are concerned, it's quite a complex process. And what you need to understand is that we're releasing inactive enzymes at the onset of digestion of protein. And what we call these inactive enzymes are zymogens. Okay, so trypsinogen here is a zymogen that is used to activate trypsin, which then trypsin is going to activate other enzymes, chymotrypsinogen, which then activates chymotrypsin. We also have trypsin activating procarboxypeptidases, which then is going to stimulate carboxypeptidases to do their job, which again here the job is to digest protein. We also have elastase and endopeptidase, which stimulate polypeptides to basically break apart into oligopeptides and tripeptides. So the end result of these zymogens activating the enzymes and elastase and endopeptidase doing their job is that we're releasing some oligopeptides, some tripeptides, which contain 3 amino acids, and some free amino acids. We also have the Brunner's glands releasing mucus at this point as well. The enterocytes, or the mucosal cells of the gastrointestinal tract, also release some enzymes. They're releasing aminopeptidases, which take oligopeptides and break them apart into dipeptides and free amino acids. Where they're also releasing dipeptidyl aminopeptidases, which take dipeptides and release some free amino acids, as well as tripeptidases are released, which take tripeptides and convert them into dipeptides and free amino acids. Again, the whole point here is that we're digesting these large proteins into smaller and smaller pieces so that we can actually absorb them across the gastrointestinal tract. In the large intestine, we find very, very little protein. We are very efficient at absorbing and internalizing protein. So there isn't going to be very much at the level of the large intestine. It is important to note that diseases like celiac disease, which is an intolerance to the protein gluten, can lead to changes in the amount of protein that you'll end up with in the large intestine. So if the gastrointestinal tract is inflamed in any manner, you'll find more nutrients in the large intestine that you wouldn't normally find in somebody who has a healthier gastrointestinal tract. It's interesting to note that the microflora also are going to produce some materials from carbohydrates and amino acids that provide us with some health benefit. So that's important to keep in mind here as well. We also have ammonia from deamination, and deamination just simply means that you took off that amine group, that nitrogen group that makes protein so special off that amino acid, and so you can release some of that, which then could be converted to urea and in some instances recirculated back to the liver so that we can recoup some of that nitrogen and utilize it to basically make other amino acids. We also have deamination of aromatic rings, which yield phenolic compounds, which phenolic compounds have been shown in a person's diet to basically reduce our risk for cancer. So how do we internalize these proteins? How do we absorb them, get them into the bloodstream? Well, most of the absorption is occurring at the distal duodenum and the proximal jejunum. And most absorption is going to occur with the proteins, these complex structures, broken down into free amino acids, dipeptides, and tripeptides. That's how we're basically going to absorb protein as. So, they're going to be transported into the enterocyte, so that gastrointestinal tract cell, using a couple of different mechanisms. The first of which is facilitated diffusion. And facilitated diffusion does not require energy. It does require some sort of transport mechanism there, but it doesn't require ATP. Now, these systems are still in the research phase, so we don't quite have all these systems down. So you may see some differences in the scientific literature as far as these systems are concerned. But the point here is that amino acids are very complex in not only their digestion, but also the transport across the gastrointestinal tract cells. So, for example, there are these specific transport systems, like the L system, which target branched-chain amino acids and other neutral-type amino acids. It's only going to target those particular types of amino acids. And in this instance, it happens to have a very low Km, which just means that we're going to preferentially absorb those. As you recall, the branched-chain amino acids are all essential amino acids, so it would make sense that we'd want a transport system that would really absorb those quite quickly so that we internalize them and use them for our protein needs. There's also the Y plus star system, which targets praline and glycine. There's the B sub naught and the plus system, which can also target most neutral and basic amino acids. So there are some very specific transport systems that will transport these amino acids into the enterocytes. We also have active transport by some of these specific systems. There's the B system, the Amino system, the X negative AG system, and the B sub naught and plus system, which target other specific amino acids and require ATP. That's what makes them active transporters, that they need ATP to be broken apart in order to get them to be internalized or absorbed within the enterocyte. So we also absorb protein as peptides in the form of dipeptides and tripeptides. So that is two amino acids put together to make a peptide, dipeptide, and tripeptides, three amino acids to make a peptide protein. 67% of protein is going to be absorbed as these peptides. They, again, have very specific transport systems to get them across that gastrointestinal tract membrane and into the enterocyte, and we're still working on determining all these systems. They do require the entrance of hydrogen ions into the enterocyte, and you have to have depolarization of the brush border membrane, which is the membrane that's in contact with the lumen of the gastrointestinal tract. And it's also dependent upon the sodium-potassium ATPase pump, so it requires ATP so that you can pump these hydrogen ions back out and get sodium back in. These peptides, interestingly, are absorbed more rapidly than just free amino acids, and as the length of the peptide increases above 3 peptides, the affinity for transport decreases. So it seems like the dipeptides and the tripeptides, as well as the free amino acids, are how we are internalizing protein. So, as far as these transport systems are concerned, it's really important to understand this particular concept, and that is the idea of competitive inhibition. And it's based off of the fact that if we have specific transport systems for amino acids, and if we have amino acids that are sharing those transport systems, then if I overconsume a particular type of amino acid, I could run into the possibility where I now have competition for absorption. And of course, as with most of these things, the higher the concentration of a particular amino acid, that amino acid is basically going to win and be absorbed preferentially over another amino acid. That could lead to a deficiency in that particular amino acid. So it's important to have a balance of all the amino acids and not overconsume one. So the main group here at risk could possibly be the bodybuilder. If they're running out and going and buying individual amino acids and perhaps overemphasizing one particular amino acid over another, then they may run into this problem. So we should be on the lookout for this with any athletes that are emphasizing protein supplements and amino acid supplements in their diet. Okay, so once we get inside of the enterocyte, So we've crossed that brush border membrane where all the villi are into the enterocyte. We actually will hydrolyze any peptides. So those dipeptides and those tripeptides will be broken apart using hydrolysis, and we release the amino acids from those peptides. So we get all these free amino acids now. Some of those amino acids are going to be used directly by the enterocytes for energy. Glutamine in particular is primarily used and preferentially used by enterocytes for its needs. In fact, it's considered a trophic factor with intestinal cells, and it's been shown quite useful in the literature for a clinical setting where you have somebody who's on an enteral or tube feed in maintaining the health of the gastrointestinal tract cells. So it can be quite useful in a clinical setting. So glutamine can be used to make several different compounds, as you see listed here. It could be used to make glutamate, which can be converted into alpha-ketoglutarate and then utilized in the Krebs cycle for oxidative metabolism to produce ATP. We can also use glutamine to strip off the ammonia group and then send that off to the liver, or keep it within the enterocyte and make other amino acids out of it, and then ship it off to the urea cycle. We can also take an amino group and add pyruvate to that, and that will produce alanine, which then gets sent to the liver and can participate in what we call the glucose-alanine cycle, which we'll cover a little bit later on. But the point there is that we can make some glucose out of it. We can also use glutamine to make glutathione, which is an important component of the antioxidant system that we have that protects us against free radicals. And we can also use it to make proline and ornithine. Some amino acids in the enterocyte are going to be used for protein synthesis. So we can make the apoproteins that are required in the construction of chylomicrons. We can make new digestive enzymes and even hormones. Transamination can also be undergone in the enterocyte. Transamination is simply when we take one amine group from an amino acid. So in this example, we have valine. We're taking off of that amino group, we're deaminating it, and we're attacking it onto what's called a carbon skeleton, and basically forming glycine out of it. We can also now transport some of these amino acids outside of the enterocyte. And where are they going to go? They're going to go into the hepatic portal vein and directly to the liver. And what's important to understand here is that there are also transport mechanisms to get these amino acids out of the enterocyte. So they're very similar to the brush border membrane that we saw that faces the gastrointestinal tract lumen. And this particular membrane is called the basolateral membrane. So at the level of the basolateral membrane, in contact with blood supply, we're trying to have transport mechanisms that get amino acids outside of the enterocyte. And so again, we've got these transport systems that are very specific for specific amino acids, and they all have different Km values. So some special notes about protein and amino acids. In research studies, it was found that if you simply just ate whole foods containing protein, and you compared that with an equivalent mixture of free amino acids, so you were getting the same amino acids, it was found that the absorption of protein was actually more rapid if you just had whole food compared with a drink mixture of amino acids that were already broken down. It was also found that nitrogen assimilation with protein from whole foods was actually better than with free amino acids floating around. So contrary to public dogma, free amino acids, at least in the literature, really has not seen any absorptive advantage in healthy individuals. There may be some clinical application for this, but as far as healthy individuals, as far as athletes are concerned, there's really no advantage here. So where do these absorbed proteins go? Well, 99% of ingested proteins are going to enter the hepatic portal vein as amino acids. So you do get some oligopeptides that are sneaking in, and they're sneaking in through the tight junctions, or they get in through transcellular endocytosis. But for the most part, you're getting amino acids that are going directly to the liver via the hepatic portal vein, and they are transported in blood in the plasma part of it because they are water soluble. So once we get to the liver, what's going to happen here? Well, about 50 to 65% of these absorbed amino acids are going to enter the liver. The branched chain amino acids are actually going to be released back into the bloodstream. And those are basically going to muscle tissue. Muscle loves branched chain amino acids, as we'll talk about. They're going to have similar transport systems to the basolateral membrane. So in other words, to internalize them and also to release them, you have to have, again, these very specific transport systems for each of the amino acids. Here are some of those systems: the A system, the glycine system. Again, very specific for amino acids. Okay, so about 20% are going to be used within the liver to synthesize new plasma proteins. And these plasma proteins, more often than not, are staying in the liver. Some of them are going to be released in the plasma, however. The substances that are released in the plasma are going to be the glycoproteins, globulins, lipoproteins, and immunoglobulins. So those are all produced within the liver and then released into circulation. The liver also produces visceral proteins, which all of these listed here are indicators that are used in a clinical setting as to protein status. So there's albumin, there's transthyretin or prealbumin, which is a better short-term indicator of protein status compared with albumin. That's more of a long-term protein status that takes about 3 weeks to turn over. You've also got retinol binding protein as well as blood clotting protein. So these are all viscerally produced in the liver, and we call them visceral proteins. We also produce acute phase proteins, which are proteins that react to some sort of stressful situation or some sort of inflammation. There's an immune response there. So for example, C-reactive protein would be an example of an acute phase protein. So they're stimulating the immune system, they're promoting wound healing. There are also these stress or heat shock proteins, HSP60 and 70 and 90, etc. There are a whole slew of these that are being studied. I would say, in the last 10 to 15 years, these have been in the literature quite extensively, particularly with exercise, because they are released with stress, but however, heat stress and oxidative stress that we encounter during exercise could possibly indicate some problems here, and perhaps in overtraining syndrome with athletes. We're not quite sure, but that's the idea, is to target some of these stress or heat shock proteins and trying to use them as evaluative markers for athletes and their health. We also produce nitrogen-containing non-protein compounds. So we use the nitrogen from some of the amino acids that we just ingested, and then we produce these non-protein compounds. Glutathione is one of those. That's part of an antioxidant system. It protects us against the free radicals. We also have carnitine, which transports fat into the mitochondria. So it's transporting those long-chain fatty acids, those really big fats, into the mitochondria, particularly in muscle tissue. We have creatine, which is being used to store phosphate for energy use by muscle tissue. And we have carnosine, which has some antioxidant activity, as well as choline, which is part of a neurotransmitter, the acetylcholine. So that's all being produced within the liver, and they've got this nitrogen component, and you're trying to produce this non-protein compound. We also have the synthesis of purine and pyrimidine bases, so we can produce DNA and RNA. That's also going on. And some amino acids in the liver are undergoing transamination, so we're transferring that amino group to another carbon skeleton to produce an amino acid. So that's transamination. So we can make our non-essential amino acids from essential amino acids. Some of these amino acids are going to be deaminated. So we're going to remove that amine group and basically be left with this carbon skeleton that we can use to make other things. The ammonia, it's important that the ammonia, that amine group, has to be disposed of, and we dispose of that amine group in the urea cycle. So the liver is producing urea, and then that urea is going to be filtered out by the kidneys, and then, of course, urinated out. If you have liver failure, this is a great time to appreciate your liver for all the things that it does. The liver failure can cause basically a hepatic coma from ammonia levels reaching so high in the blood that it causes the brain to shut down. So thank your liver for everything that it does, including deamination of amino acids. So the stuff that we have left over, as I mentioned, is what we call a carbon skeleton. They're also called keto acids or alpha keto acids. And this is basically an amino acid without the amine group. And the neat thing about these carbon skeletons is that we can make other things out of them. So we can actually use the carbon skeletons for energy. We can make some glucose out of them. And in fact, the liver can use these carbon skeletons for about half of its energy needs. So this is basically where you're going to see the most catabolism of protein is going to be within the liver. So about 10 to 15% of total ingested protein is going to be used by the liver for ATP production. We can also use those carbon skeletons, as I mentioned, to make glucose from what we call gluconeogenesis. About 50 to 60 grams of glucose will be produced for every 100 grams of protein that I utilize for gluconeogenesis. And there are some amino acids that are more glucogenic than others, and they're listed here. Some other things that are produced, ketone bodies can be made from these carbon skeletons, and we call some of the amino acids that make these ketone bodies in this process ketogenic amino acids. So lysine and leucine are very ketogenic amino acids. We also need these amino acids for cholesterol production, and it's produced in a roundabout way. So you basically need leucine to make an enzyme, HMG-CoA reductase, to facilitate the process of making cholesterol. So HMG-CoA reductase is an enzyme that catalyzes the reaction that makes cholesterol. As far as fatty acids are concerned, we can take that carbon skeleton and we can produce fatty acids out of them and then store that excess amino acid that we ate as triglyceride in adipose tissue. Any amino acids that are not taken up or released by the liver into the bloodstream actually form what we call an amino acid pool. And this is a very, very small pool, only about 170 grams of this. And mainly like the liver, a little bit in the blood, and some in muscle tissues where you're going to find this amino acid pool. But it's found in a variety of tissues in the body. So it's going to be the primary source of amino acids for extrahepatic protein synthesis. So when you talk about protein synthesis, those amino acids are being drawn from this amino acid pool. It remains relatively constant based on the input of protein, so the consumption of protein, as well as some of the losses of protein. What's interesting about this amino acid pool is that we have far less essential amino acids than non-essential amino acids in the pool. And the essential amino acids that we find in large amounts in this amino acid pool are going to be lysine and threonine. As far as non-essential, we see a lot of alanine, glutamate, aspartate, and glutamine. In fact, out of all of these, glutamine is found in the highest concentration as far as this amino acid pool is concerned. Some of these amino acids will be used for catabolism of body tissues. So basically we can catabolize some of these proteins and release those amino acids and then use them for energy when there's an inadequate caloric intake. So if we're not eating a lot of calories, it goes without saying that your protein will probably be a lot less, and we'll start utilizing body tissues like muscle for the release of those amino acids so that we can utilize them for energy production. The young child that's down here in the bottom right-hand corner of the screen is suffering from what we call marasmus, which is a protein energy malnutrition a condition where overall caloric intake is quite low and thus protein is quite low. And as you can see, he's pretty much the skin and bones because of all the muscle wasting from catabolism. Within skeletal muscle, again, we're using some very specific transporters to get the amino acids within muscle. We're preferentially taking up branched-chain amino acids, or BCAAs for short. It's stimulated by insulin, so the transport proteins for branched-chain amino acids are stimulated by insulin, and we're drawing in those branched-chain amino acids from blood into muscle. And muscle tissue loves to catabolize these branched-chain amino acids for energy. And we'll talk a little bit more about that once we get to the metabolism section of the course. We also are going to stimulate protein synthesis in muscle tissue as we're bringing in amino acids. The kidney also utilizes amino acids. It may be a part of the gamma-glutamyl cycle transport system. The kidney can do some gluconeogenesis, so you can take some of these amino acids, strip off that amine group, left with the carbon skeleton, and use that carbon skeleton to make glucose. We're also going to rid the body of nitrogenous waste here, so the kidney is going to relieve some of that protein load that we've just ingested as urea, some as creatinine, some as uric acid, and of course ammonia. We have very small losses from the kidney because, remember, protein, it is a primary macronutrient, very, very important to us. It's ubiquitous in the human body, so we try to minimize the loss of protein. So about 0.3 to 1.0 grams per day as far as loss via the kidney. The brain also utilizes amino acids, and we have transport systems very specific for neutral, dibasic, and dicarboxylic amino acids. We use these amino acids to produce neurotransmitters, hormones, and neuropeptides. A great example that we'll talk a little bit more about later is the production of serotonin from tryptophan, which then leads to melatonin production, which then could make you sleepy. And so the idea here is, as we'll talk about with the central fatigue theory, is that if we consume branched-chain amino acids, we can actually slow some of this process down according to some of the literature. So some special notes about protein. Essential proteins, as far as the amino acids are concerned, if you don't have those present, will shut down protein synthesis. So if you don't have the building blocks to build whatever it is that you're trying to build, you're basically not going to build that particular protein. And so this is where protein quality really comes into play, and we'll discuss this later on in the course. Athletes really need to be cognizant of protein quality, because if they're not getting all the essential amino acids, then that could consequentially, shut down the production of protein synthesis, or proteins by protein synthesis. So we don't want to affect transcription and translation of proteins. So what regulates protein metabolism? Well, a good part of it is going to deal with dietary intake. So that's really going to influence the metabolic processes. If you have an inadequate intake of protein, that will actually suppress catabolism. So we don't really utilize protein for energy, at least we try not to, when the intake is lower than it should be. As far as when we have sufficient intake of protein, then we actually stimulate catabolism, because we can spare that protein and we can utilize that protein for energy needs. We also have glucagon and cortisol, which both are going to stimulate catabolism. So if calories overall are insufficient, then we are going to release cortisol and glucagon, which is going to stimulate catabolism to release those amino acids so we can utilize them for ATP production.